Hello, everyone, and welcome back to 10 Seconds. Imagine walking into a massive library containing 1.7 million different books, but they are all just piled haphazardly on the floor. To find anything, you'd need a system for organizing it all. Earth is exactly like that library, and those 1.7 million books are the distinct species of living organisms we share this planet with. Take a specific historic text like Charles Darwin's On the Origin of Species. Without an index or a filing system, locating this single book among millions would be difficult. Biologists face the exact same problem when studying life, which is why a rigorous system of classification is necessary to identify and track organisms. Life isn't distributed evenly. We have areas with exceptionally high concentrations of biological diversity. We call these biodiversity hotspots. In India, the Western Ghats, the Himalayas, the Indo-Burma region, and Sundaland are incredibly rich zones packed with diverse life forms. Many organisms in these hotspots are what we call endemic species. For your exams, remember this strict definition. An endemic species is restricted to one specific geographical region and is found naturally nowhere else. The Nilgiri tahr and the lion-tailed macaque are perfect examples of species endemic to India. We can see how specific environments dictate where species live by looking at the Pakke Tiger Reserve in Arunachal Pradesh. Scientists tracked 4 different species of hornbills there. They found that each specific species distributes itself strictly based on the size of the available nesting trees and the exact types of fruit growing in that specific part of the forest. Scientists use classification as a fundamental tool to map these interconnected life forms, providing the biological framework needed to understand how entire ecosystems survive. This timeline flowchart maps how our classification systems evolved as our technology improved. Aristotle started by simply grouping animals by habitat, land, water, or air. Then came Linnaeus with 2 kingdoms, followed by Haeckel and Copeland. Finally, we arrive at the definitive system we use today, R.H. Whitaker's 5-kingdom classification. Whitaker's first group is Kingdom Monera. These are the simplest single-celled life forms on Earth, like bacteria and cyanobacteria, the organisms responsible for producing Earth's early oxygen. Their defining structural feature is that they do not have a proper membrane-bound nucleus. Next is Kingdom Protista. If you do a hay infusion activity in your lab, you'll see these microscopic organisms swimming around. They are also single-celled, but unlike Monera, protists, like the amoeba, do possess a true membrane-bound nucleus. The third group is Kingdom Fungi. These organisms have cell walls made of a tough substance called chitin. Ecologically, they act as saprophytes, breaking down dead organic matter. This biological mechanism is exactly what makes large-scale mushroom farming a viable economic application today. Moving to Kingdom Plantae, we can track plant evolution like a staircase. At the bottom are Thallophyta, simple algae that live entirely in water. The next step up is Bryophyta, like mosses. We call them the amphibians of the plant world. They made the evolutionary leap to moist land, but they still strictly require water to reproduce. The climb continues with Pteridophyta, like ferns. They developed dedicated vascular tissues, xylem and phloem, to transport water and food, though they still don't produce seeds. To survive cold, dry conditions, gymnosperms like pine trees took the next step, developing naked, exposed seeds. At the top of the stairs are angiosperms, the flowering plants. They use flowers to attract pollinators and enclose their seeds inside fruits for efficient reproduction and dispersal. Early Indian texts, like the 17th century Hortus Malabaricus, systematically documented these complex plant traits long before modern genetics. The entire evolutionary ladder of the plant kingdom is driven by a single objective: solving the challenges of living on dry land. Every new trait was a biological mechanism to balance growth, nutrient transport, and reproduction without relying on constant water. Kingdom Animalia follows a similar progression of structural problem-solving. At the base are porifera, or sponges, just multicellular bodies covered in pores to filter water. Step up to Cnidaria, like the hydra. They develop the first true tissues and tentacles to catch prey, but they are limited by having only a single opening for both taking in food and removing waste. Platyominthes, the flatworms, solve the movement problem by introducing bilateral symmetry, a distinct left and right side. Nematoda, the roundworms, solve the digestion problem by evolving a highly efficient tubular body design with two completely separate openings, a mouth and an anus. Annelida, like earthworms, induce segmented bodies and true internal cavities for better movement. Pause and ponder this for a moment. Why does the hard exoskeleton of the next group, Arthropoda, which includes insects and crabs, provide a massive survival advantage over those soft-bodied worms? It provides rigid protection and prevents water loss, allowing them to conquer dry environments. The final major groups include mollusca, which protect their soft bodies with shells, and echinodermata, which use hard internal skeletons. This brings us to the critical transition into protochordates and vertebrates. Their defining feature is the arrival of the notochord, the flexible rod that acts as the foundation for a true backbone. From a flatworm's bilateral symmetry to a mammal's rigid backbone, every animal feature is a distinct structural adaptation. These physical changes accumulated over vast spans of time, allowing organisms to hunt efficiently, evade predators, and survive in extreme environments. To keep track of all this complexity, scientists use a biological address system. This inverted pyramid represents the hierarchy of classification. You can remember the order from broad to specific with a simple mnemonic. Keep pots clean or family gets sick. That stands for kingdom, phylum, class, order, family, genus, and species. To prevent confusion across different local languages, Carolus Linnaeus introduced binomial nomenclature. There are strict rules. The scientific name always has two parts. The genus comes first and is capitalized. The species comes second in lowercase. And finally, the entire name must be italicized in print, or underlined if handwritten. As genetic tools advanced, scientists started comparing organisms at the DNA level. In 1977, Carl Woese proposed the three-domain system: bacteria, archaea, and eukarya. The genetic data proved that microscopic life was actually far more diverse and complex than the old physical classification systems could see. All this classification directly informs how we protect life today. This is an aerial view of the Fumdes, unique floating masses of vegetation and soil on Loktak Lake in Manipur. This specific, fragile habitat supports a highly specialized web of life. When a habitat like the Fumdes disappears, the endemic Sungai deer that relies on it goes with it. This loss fractures the real-world biological connections we've been mapping through classification, proving that protecting these categories is a matter of life and death for the species involved. Let's test your recall. We talked about the amphibians of the plant world earlier. In the comments below, tell me what plant group that is, and compare them to animal amphibians. What survival problem do they both share? Don't forget to like this video, share it with your classmates, and subscribe to 10 Seconds. Keep learning, and I'll see you in the next one.